Science. Technology. Medicine. Health. These four things make the world go round. Without them, we couldn't exist. This is the Monday Science Podcast, a weekly show bringing you the latest research and news in science, technology, medicine, and health, answering your questions or finding experts in the field to answer them. Your host is a pharmacist, an award-winning scientist. She leads her own research group and is the founder of King's College London Fight the Fakes. A tad bit on the qualified side. Welcome to Monday Science. Here's your host, Dr. Bahijja Raimi-Abraham.
I was initially going to start the episode with a I don't know how professional that was, but yeah, hello, welcome back to Monday Science. We have a new intro, we have a new outro. Things are changing around here, but for the better. What did you think? Was it intense? Did you feel excited? Are you excited for today's episode after that intro? I hope so, because it feels like it's been a while since I actually spoke properly on the podcast. It has just been a hectic past few months. So for those who may be new to the podcast, my name is Dr. Bahijja Raimi-Abraham. As the intro let you know, I'm a pharmacist and a scientist, and I have my own research group and so forth. But one of the things about being an academic is that you have your research, and that's great. And then you also have teaching and admin, which, depending on what you like, can be great, cannot be great, but whichever way, it can be very hectic. And we've just come out of a very, very busy time with student exams, marking, editing. Sorry, editing is for the podcast, obviously, marking and feedback and all things intense. And that's been ongoing since around May, actually. So roughly when we had the 10 year, 10 years. What is wrong with me? I think the intro has thrown me off. No, we had our 1 year anniversary on the 10th of May, and that was sort of around the very busy period for me professionally, which is why having the break was great. And this goes back to, you've got to look after yourself. Your mental health is so important. And so it's been very, very hectic, and I have really missed the podcast. I've really missed talking to you, and I hope you've missed listening to me. But obviously we had some episodes, we started back, and that's good. And yeah, I guess I'm just trying to get back to some sense of normality. I am apologizing if this is a bit of a waffle. I feel like I'm a little bit out of practice. It's been a little bit of time. So marking and all that joy has finished for round one, and then we have another set of marking in August for the resits. So but it's usually less intense. It's been I'm still waffling on about marking. You can tell that this is going to be a little bit of an outlet. But anyway, we have an interesting episode to get through. But I will just talk about marking briefly. It's just a very intense time. And this is the third time marking online. And I don't know if anybody else is feeling this, but whilst, yes, we use our laptops and computers and 2 screens, 3 screens, 4 screens, if however many screens you need, but it just gets so tiring just always staring at a screen. I'm actually doing an eye test. I'm concerned my eyes, my prescription has probably changed once again just from staring at the screen. But yeah, so when it comes to marking, we usually have very tight deadlines, and it's just been very intense. But anyway, enough waffling about marking. Welcome back to Monday Science. So happy to be able to speak to you all today. Hope you're enjoying the amazing, well, those of you in the UK, hope you're enjoying the amazing weather that we know is only probably going to last one more day before we are welcomed with rain. I also feel we're usually a day or two away from sleet and snow. But anyway, all that aside, I hope you're enjoying the really nice weather wherever you are in the world, whatever it is. Let's get on to today's episode. So, we're going to start off with our Monday Science Person of the Week. This goes to Rachel Ignatowski, and she is a New York Times best-selling author and illustrator who creates work inspired by history and science. Rachel has published a book called Women in Science, where she discusses the lack of representation of women in STEM and credits and acknowledges the work of 50 women in STEM who have failed to gain recognition despite their work, discoveries, and dedication. So amazing work that Rachel is doing there. I've got two interesting news updates today. So one, the title being apparently Viagra can teach us a lot about treating rare diseases. So for those of you who don't know, Viagra was originally developed to treat angina, so that is a heart condition. But early clinical trials showed that it wasn't actually having enough of an effect to make it a commercial success. And we do have to remember that there is healthcare is a business in its own right, and when drugs and medicines are being made, it's for the end user, who's the patient, and there is a commercial element obviously there. So the scientist David Brown, he was working for Pfizer at the time. His new drug, which I don't think they called Viagra, it's sildenafil is the active ingredient, and initially targeted for angina. It wasn't doing well in clinical trials. Pfizer said, "Look, you've got 3 months to turn this project around." And a few weeks later, apparently, David Brown heard some very unusual news from a group of Welshmen involved in the clinical trial, and they were experiencing more erections than usual. I'm interested to know what the baseline would have been for that. So, realizing that, hold on, may not be good for angina, but actually could be good for something else. Pfizer switched the clinical trials to focus on erectile dysfunction. And so we know about the success of Viagra and the impact it made in society and all that. And so David Brown now wants to repeat this trick to find new treatments for rare diseases with his startup Helix. So a couple of years, was it last year? Time merges into one, but a few years ago, I attended this medtech conference, and there was there was a representative from a company called Helix. And so David Brown co-founded this company Helix in 2014 with somebody called Tim Gwalliams, and it's a Cambridge startup which uses artificial intelligence to identify existing drugs that have already been through clinical trials and then repurposes them for treatment. And we've probably heard over the last 17, 18 months about drug repurposing. It's a cheaper way to find new treatments for diseases. Also, it means usually that the drugs have gone through necessary clinical trials for safety, general safety, and so forth for whatever condition it was initially licensed for. And so when you're repurposing, you're just looking to see what doses would work and if there's efficacy in the disease, the treatment, the condition at hand. Um, and it was just so fascinating to learn about their work. The idea of the company came about for a discussion between Gwalliams Brown and somebody else called Nick Sarayo. They had two sons, and their sons had a rare genetic disease, and so they've come together. And so right now they're looking to see, if Viagra could have possible sildenafil. Let's use the active pharmaceutical ingredient that is called API. Sildenafil can have potential in rare diseases. Now, after this is just an additional piece of information. So sildenafil, which is Viagra, there are other drugs that are used for erectile dysfunction. Tadalafil, which is I can see the thing in my face. I can't remember the name, but they're different drugs that are used, and they've also found use in pulmonary hypertension. So repurposing of this therapeutic class of drug does go on. The second piece of news that I wanted to share with you guys today is a little bit different, and just because of time, I've put the link to the article in the episode description. The article is titled "It's Time for Alexa, Google Assistant, and Siri to Swear." And it's a really interesting article written by Jeremy White, highlighting that voice assistants need to speak to us in the same way we talk to them. Not saying that everybody goes around swearing, but it's to try and make these voice assistants be more natural and human like. A very interesting article. Strongly recommend you have a read. So, on to today's episode. Well, today's episode, or this week's episode rather, is sponsored by the Black Dementia Company. The Black Dementia Company is a UK based service that was set up with the aim of meeting the needs of persons living with dementia and their friends, relatives, and caregivers across the world. Their hope is that through their products and services, they can contribute towards improving the lives and well being of persons living with dementia, including and especially persons living with dementia from the global African and Caribbean community. Their products are available on their website, theblackdementiacompany.com, and also on Alzheimer's Society Shop's website. All details are in the episode description. In this week's episode, episode 80, I'm answering the question, how do scientists study the brain? With my guest, Professor Alysson Renato Muotri, who is a professor at the Departments of Pediatrics and Cellular and Molecular Medicine at the University California, San Diego. We have Professor Alysson Muotri. How are you today?
I'm fine, thank you. Thank you for having me.
Good. Thank you so much. I was just saying my excitement for having you on the podcast, being a fan of your work, having read your work, and even had a whole episode dedicated to the topic of brain organoids and sentient abilities. And we're going to talk all about that today. I'm going to try not to fangirl, so sorry. So firstly, if you could start off with telling us a little bit about yourself, please.
Yeah, so I am a faculty here at UC San Diego, and I'm actually from Brazil, in São Paulo, which is like a big city. And after I finished my PhD in human genetics, I moved to California for a postdoc in neurosciences and stem cell biology, and decided to stay. And I established my lab in 2008. And since then, we have been using stem cell technologies really to mimic human brain development for different applications.
Wow, that's really interesting. I like San Diego. I've only been there once. It's meant to be like the best city in the world, I believe.
Yeah, I agree with you, and that's why I decided to stay.
Yeah, I can imagine. It's very, very nice. So do you have a favorite song at the moment that you can share with our listeners?
Favorite song. That's a good point. I change it all the time. Let's see. I think recently I've been listening to Dua Lipa.
Oh, yeah, I've heard of her.
She has like some really nice songs, and I play in the radio all the time, and I like it. Yeah.
Nice. Okay, so that's a good recommendation. Thank you. This is just questions that I'm asking because we have a Spotify playlist for all the songs that our guests have recommended. So we'll add some Dua Lipa on there. And then if you have maybe a film or a book that you could also recommend, you don't have to do both, but one or the other.
Yeah. So book. Recently, I've been reading this one. Let's see if I can.
Let's see. The brain. The brain inside out.
The Brain from Inside Out, and from Georgi Budzaki. He's a really famous neuroscience, and the book is just amazing. So I'm a big fan.
Oh, thank you. Good recommendation. I need to actually start reading some of these books. The list is getting very long. So thank you for those recommendations. So you gave us a little bit of an overview of your research. So how did you become interested in studying neurodevelopment disorders and autism specifically? And then we're going to get onto organoids. I'm going to try not to overtake that.
My interest was very academic. I was always fascinated by the human brain and the evolution of the human brain and what makes us different from other species. One thing that when you start studying that, you realize that the human brain has evolved to become very social. So we are social. We are a very social species. Even compared to other species, we have some kind of a social relationship with each other that is just amazing. You might say, well, I mean, bees do that as well. They have like a very systematic way to organize in society, but you don't see like bees trying to cut the head of the queen and take the power. So we humans, we do that all the time, and it's really about our frontal cortex that has evolved to accommodate these sophistication social abilities. So I decided to study that. How did the human brain become so social? And one way that geneticists used to approach these kind of questions is to look for mutants. So we started looking for when does the social brain does not work properly. So that turned me into autism and other social disorders. So I become interested on autism, and then I realized how difficult it is to study autism because it actually starts in uterus. And I cannot use animal models, or animal models are not the best models for this kind of conditions, because it's a human condition. So there is no autistic mouse model out there, or any monkey autistic individual that I could use. And more than that, I mean, autism starts in uterus, so it's inside the womb, and it is hard to experimentally access that. So I cannot just ask for a pregnant woman to join my research. And by the way, I'm going to stick some electrodes in your fetus. So it's not going to happen. So we have to find ways to study that using like a very different approach. And that's where stem cells comes in. I can use now stem cells that can be derived from people with autism to reprogram their cells and recreate brain development outside the uterus. So the technology is getting better and better all the time. And now we have good protocols to create what we call these brain organoids. So a brain organoid is a cluster of cells, and can be up to like 2.5 million neurons in there, that self-organize in a three-dimensional structure that resemble these very early stages of neurodevelopment. So that's a very nice tool, because I can create as many as I want inside my lab, and I can study them, and I can compare from individuals with autism and neurotypicals, and ask what is the difference between them. So this is the bread and butter of my research.
Fascinating. So interesting. So as I mentioned before, I have an interest in organoids and spheroids, but my interest only, I mean, you mentioned something about when you are wanting to research something, but you don't have access to it in the lab, essentially, And so my journey into understanding organoids and spheroids was, well, I say understanding, I'm still trying to understand. Organoids specifically was because I have an interest in malaria research. But as you know, when you're setting up your lab, to get sort of the ethical approval, the animal license, all those various different things. And in particular, in your case, where there is no animal model, that would be suitable, you start to think, okay, what about some in vitro technologies? And so one of my colleagues, he's been doing some organoid work for intestinal models. And then I don't remember, I think there was a Nature News article talking about organoids and sentient ability, brain organoids. And I hadn't even thought about the sort of, it's a limitless technology, because if you can make an organoid, you can make it for any organ, really.
Yep.
And I think that's quite fascinating. My interest is more around scaffold development, so trying to optimize the scaffolds to see how can that influence differentiation and just the cells of the organoids growing in general. So yes, as I mentioned, I'm really trying to not fangirl in this moment, but your news feature was Can Lab-Grown Brains Become Conscious? And then it was episode 25, so that was where I discussed it. But then also within that article, you talked about how you temporarily pivoted aspects of your research to study COVID-19 using your brain organoids. And this was to test how the drugs had been performed. And it's summarized in another article I'm going to put in the episode description. So aside, and you gave a nice insight into how you sort of decide like why organoids are important, but how did you even get there? I don't know. The only way I could put it is, how did you get there from your stem cells? Just talk me through the process, please.
Yeah, so I will start from the beginning. So the first thing that you need to do is to find volunteers or subjects for your research. And you start by consenting people. So there is a consent form, and you ask for live cells. So that's the raw material. We need to start from live cells. So yeah, I mean, most of the subjects in my research, they have someone that is affected by a neurological condition in their families. And then, of course, I mean, they will do anything to help them. So they understand the research, they explain what we are trying to do, and they donate cells. Most of the time, these are skin cells or dental pulp cells from the milk tooth. And we can isolate these cells in the lab and reprogram them to become pluripotent stem cells. So this would be something similar to an embryonic stem cell. So we go back in time and reprogram those cells to lose any identity that they might have. So after the reprogramming, they are no longer a skin cell or a dental pulp cell. They are a naive cell with no identity. And from that stage is when we can add factors to those cells to instruct them to become whatever cell type we want. So since they have this plasticity, my lab has been specialized on learning how to drive the cells to different brain regions. So we know what kind of factors the cells respond to become a neuron or to become a glia cells. So that's what we do. I mean, we let them to fate committed by adding those specific factors, and we do it in a very transient way. We just expose them for a short period of time, and then we take it out and let them go. And that's the amazing part that I think is almost a miracle, because it's all genetic program. So they know what to do. They start to differentiate, they start to migrate, and they start to form these little brain structures that are very similar to the developing human brain. I think this is fantastic. And I'm so glad that the cells can do that by themselves. In the early stages of this process, we are all wondering if we have to instruct the cells to go to specific places. I'm so glad we don't have to do that, but we rely on the genetic programming of the cells itself.
Fascinating. And yeah, that's the novelty of the fact that the cells do it on their own because of the processes and however you direct them to do that. I think that's really fascinating. And I guess, sorry, this is just a little bit, well, it's within the topic. As you were explaining how you do this, so you actually start off with live donor cells. And then, because I think from, again, limited understanding in this area, but growing, people tend to just get their cells, ordered from whatever company, and then they use that. So I think that's probably what even makes yours so close to life, human-like, because you're actually using real patient samples. As you were describing everything, do you think there's a risk that this technology could be used in an unethical way?
Yeah. And this is true for any new technology, and especially on stem cells. That's why we are always in touch with our IRB board. So these are a group of people who oversees the research and agree if the research would be performed in an ethical way. And of course, they help us and guide us through this process, so we are not doing anything that is non-ethical or borderline. So we try to avoid that. But that's why our consent form is really explaining everything that would be ever done with those cells. And we mention all kinds of things. We might genetically manipulate the cells. We might send to a colleague to explore other things. So when they sign, they agree that these are all possible outcomes. And some people decided not to sign or not to participate in the research, and that's fine too.
Yeah, I think that's this whole conversation about research integrity and research misconduct and all these things. This becomes even more important when you're working with something that is so new but has such potential. So if we're going back to the brain surrogate materials, and we compared brain organoids to ex vivo tissue and chimeras, which, do you mind also maybe explaining what chimeras are? But which would you say is the best out of all those three options?
Yeah. So first of all, I mean, a chimera is a mix of species, and people generate chimeras by transplanting human cells into animal models. And there is really no best model. I mean, each model has pitfalls and has advantage, and I think it really depends on your biological questions. For example, organoids in vitro are not vascularized. So that's why they grow up to a certain point, which is 0.5 centimeter in diameter. But after that, I mean, since they have no vascularization, there is no way for the cells in the center of these organoids to receive nutrients and continues to grow. So eventually they will die. So that's a limitation of the in vitro system. So some people are transplanting organoids inside animals, creating these chimeras. So the animal model will use their own vascular system to vascularize the organoid, in the same way that the cancer does. I mean, they attract the vascular system to inside the cells. So that you solve a problem, but you create another one, because now you have the blood system of the animal circulating through the organoid, and sometimes this is not good. Also, you touch some of the ethical concerns. I mean, we don't want the animals to suffer. So there is a certain point where these organoids will start growing to make the animal uncomfortable. And so there is a limitation there. So the advantage that you have by vascularizing the organoid might not be as good, because the organoid cannot grow that much. So it's a balance. You always have to keep in mind what is the question that you are trying to answer, and find the best biological tool to answer that. Sometimes it's a chimeric animal. Sometimes the in vitro system can do as well.
Yeah, the vascularization is something that I'd read up about, and I was thinking, oh, maybe the scaffolds could have some advantage, perhaps. I don't know. Let's figure that out. But yeah, that's really interesting. And I forget that chimeras involve interspecies. And I think when it comes to animal research, people can be, from the outside looking in, it's like people assume wrongly that in science we don't care about the animals and their care, but actually that's the whole purpose. You're very mindful of the health and the I guess the comfort of the animals as well, right. Isn't it? Okay, so how are brain organoids different from actual human brains? So with the brain organoid, is it literally a mini brain? Does it have all the little components, or does it have the core parts of what you're trying to test?
Yeah, that's a great question. And I'm not a big fan of this nomenclature, mini brains. I think the media uses to attract the attention of people, and that's fine. But in the scientific world, we know that we are quite far from the human brain, and in many ways, even number of cells. So the human brain has 86 billions of neurons, and we are talking about 2.5 million of neurons in an organoid. So that's a bee brain, very different from the human brain. Also in the organoid, we don't have all the cell types I mentioned. We don't have vascularization. Sometimes we don't have the immune cells that is also present in the brain. It's not connected to the other tissues in the body, so it's not an organism. It's not experience anything because it's inside an incubator. So all of that are limitations. But yeah, I mean, they recapitulate some of the early stages at the cellular molecular level. We recently show even at the electrophysiological network level, they can be very similar to the human brain as well. So it is a technology that is improving over time, and we think that some of these limitations will go away. Some of these limitations might not even be necessary to answer the question that we want to answer, but that's it. It's a balance. I see it that some people are super optimistic about the organoid and they hype things, and other people are very pessimistic about it and said, oh, it doesn't mimic the human brain the way it should. But that's true for any other model. So that's it.
Yeah, no, it's so interesting you say that, because I think, as we know, there's always two sides and two different opinions to one technology and one innovation. But I really like how you keep mentioning the fact that it's about the question that you have, and can that tool help you answer that question? Because it's very easy to just be like, I'm doing this, and you don't know what your question is. So I think that focus on the question, and for now, the brain organoids can help with solving that question. On the topic of that, do you think, do you feel, and this is probably a very cheeky question, but do you think, what do you think is the buzz about it? Do you think this organoid thing is a buzz, and what could be the next buzz that every move? Because remember, there was a time where it was like, ooh, 2D cell culture, amazing. Now 3D cell culture, oh my gosh, it's amazing. And then now we're on organoids and spheroids. So is it a buzz? Is it just a phase, and it's the new buzz research thing? And if so, what's the next thing?
Yeah, I mean, for some of us who have been in the road for a long time, we know that there is ups and downs on the technology, and everybody jumps on the new technology. And sometimes, yeah, the new technology brings some advantage. You mentioned like the 2D traditional culture, it doesn't have the same three-dimensional structure as the organoid. So definitely there is an advantage to the organoids. And maybe, I mean, what I feel that the next thing is at least for the brain, would be the creation of neural circuitries. So you can now start teaming up different pieces of organoids to create the circuitry that you want. So it's no longer just an isolated part of the brain, but now it's connected to different parts of the brain. And people have slowly making advances in that way. The other thing that I think it's going to be like the next big thing is the input and output on these organoids. So as I mentioned, they're not receiving any stimulus. They're not providing any information moving forward, but we can add that. So there are a couple of people thinking about, I mean, how can this be done? I mean, can we incorporate photoreceptors? Can we incorporate pain receptors? Can we start inducing some kind of information into these organoids and see how they process it? I think that's going to be very important, and it will bring us some new insights about fundamental questions we have about the brain. For example, how the brain learns. We, as a newborn, we start learning just by watching people doing and imitating. That's innate. That's intrinsic. I mean, how that happened, we have no idea. So that's, to me, such a fundamental question that we still don't know how it happens. And I think the organoids, because it's a very reductionist model, will allow us to dissect the molecular and cellular components to learn how that happens. So yeah, I mean, long answer for your question, but I think that's amazing. That's the future.
Amazing. It sounds so futuristic. Because actually, when you're talking about pain receptors and photoreceptors, so going back to the famous article, sorry, because what I don't want to say freaked me out. I've said it freaked me out a little bit. The talking about the consciousness, because as you were saying, all these different things, if you're having the organoids having pain receptors, having feeling or something like that, then surely that then opens up other ethical questions. Yeah. Because if you're creating something with not emotion but sensory and because I remember the article you talk about, there was a conversation about consciousness, and it was, correct me if I'm wrong, but in some of the studies the consciousness was on the similar level to like preterm babies or something like that. It wasn't adult consciousness, but again, as a lay person, you hear consciousness and you think, oh, they're going to talk to me soon. Except it doesn't have a mouth, but so yes, I just want to talk a little bit about the ethics, because uniquely with brain organoids, I think the ethical conversation, and again, please, correct me if I'm wrong, from an outside perspective, there are more ethical considerations, I feel, compared to other types of organoids. So my interest in liver, and as it relates to malaria, it's different, it's just the liver's gonna do some stuff. It's not really like, we'll be fine. So yeah, please, could we just talk a little bit more about that? Like, what would that mean if you can? And actually, maybe if we can start off with the consciousness side of things, could we just delve a little bit into that, and we can talk a bit about pain?
Yeah.
So yeah, floor is yours.
All right. So yeah, if you have asked me that question, can these organoids acquire some kind of consciousness at different levels? We know that there's different levels of consciousness. Can they acquire something? If you have asked me like 5 or 10 years ago, I would say no, absolutely not. And there are still a couple of colleagues of mine who think no, that's not going to happen. But I'm unsure now. I think that as we develop better and better organoids, more complex organoids, there is a possibility that they will eventually reach a level of consciousness or self-aware, which is going to be very hard to prove that they are in that state, even because we are not, scientists does not agree on the consciousness definition. But there is a couple of tests that we and others are thinking that we can perform with the organoids. Will not answer as yes or no. I mean, it's not black and white, but will give us some hints if they are reaching something close to a consciousness level. And I think that's inevitable because we are creating better and better organoids, and the brain eventually acquires that. We know that's what the brain does. It will eventually acquire consciousness. So that's why I think it's inevitable. So the question is, I mean, I think there are two things here. One is the public perception, right? And the public, they freak out when we talk about brain organoids, but it's not as much about heart or liver. So the brain really have a different causes of different feelings in people. The other thing is, if we really get to that level where we feel that, well, I mean, they are now showing like different frequencies in oscillation that are similar to the human brain. If you treat them with anesthetic, these oscillations goes away, which is very similar to what happens to the human brain. So if we get to that level, we might have to pause the research and discuss what is the ethical implication about that. And most likely what's going to happen is you're going to have to create a moral status to these organoids, something that we don't have right now, but in the future we might have. And similar to animal models, and by the way, all the animals are conscious and we still use them for research. So I don't think that will block the research. It's just that it will create a set of rules that we all have to agree to follow. For example, how should we dispose these organoids after we use them? How many organoids should we grow to answer a specific biological question? So these are the same kind of questions that we do it when we work with animals. And I think it might be to a point where the organoids would be in the same level.
Yeah. Public perception of the brain, you're right, actually. That's why it seems, because when you made the connection to animal models, I completely forgot that obviously the animal has a conscience. I just completely blew my, it slipped my mind. Which other organ do you think has a similar, can get a similar reaction in comparison to the brain? So you're right, the brain, when people talk about doing any form of work on the brain, you think, oh my gosh, you've got to be careful. You've got to think about ethics, things like that. I can't think of another organ that has a similar. I don't know if you had one.
Yeah, it's funny. As a scientist, I see the brain similar to the liver. But again, I mean,
there was me playing down the liver. Oh dear. Oops. Now let me listen. Sorry.
Yeah, no, but the society changes over time. I mean, in the past, we used to believe that the center of being human was in your heart. And that's why we still say, I love you with all my heart. You don't say, I love you with all my brain. So it has changed over time. I mean, how we add value to different tissues. And I think in the future, we're going to realize that in tissues, independent tissues, I mean, they have no value. The value is when they are together. And some of the philosophers, they actually believe that even consciousness will only happen if you have a body. So without a body, maybe you have no conscious. That's one of the ideas. I'm not necessarily agreeing with that, but that's one of the perceptions, one of the views.
Amazing. So why for you is the liver comparative to the brain?
I mean, just because, I mean, the tissue is there to perform certain functions, right? The liver has some functions similar to the pancreas. And I think my comparison is when we get to the functional level. And I'll give you the example of the pancreas, for example. We had pancreas organoid for a long time, and only now we have a pancreas organoid that produces insulin. So now you have a functional pancreas because it's doing what it's supposed to do. And I would argue that maybe we do have that for the liver, but we don't have that for the brain yet. Because to get to that level, the brain must organize itself and coordinate all other body tissues that we just don't have it at this stage of the technology.
Really amazing. So with the, sorry, I'm completely deviating. With the organoid pancreas that's secreting insulin, could that potentially hold promise in transplanting into diabetic patients?
Yeah, that's exactly where I think people in this field is moving to. Yeah.
Fantastic.
You've been listening to the Monday Science Podcast, a weekly show bringing you the latest research and news in science, technology, medicine, and health. We hope you've gotten some useful and thought-provoking info from the show, and we hope you had fun along the way. We know we did. We'll be back soon, but in the meantime, hook up with us on our website at www.mondaysciencepodcast.com. Shoot us an email at info@mondaysciencepodcast.com. Find us on Instagram, LinkedIn, and YouTube at Monday Science, and access episode summaries at mondayscience.medium.com. See you next week on the Monday Science Podcast.